This is a Year 8 microbes video. We did this practical where we were looking at growing microbes. Well, sometimes we call that culturing microbes, and this time we were culturing bacteria. So this is actually a GCSE required practical, so you'll come back to this again when you do GCSE biology. And we looked at trying to see the difference between alcohol hand gel and washing hands. Which one is most effective? And it's really topical at the moment, okay? Do we want to wash our hands with soap and water and then dry them, or do we want to use an alcohol hand gel? So we had our Petri dish, which was completely clean. That means we say that it is sterile, so it is prepared in a clean environment and it's sealed in bags until we use it. We had it filled with this stuff called Agar jelly. It's really rich in nutrients, so full of nutrients to help us grow our bacteria, providing a food source for it. And that was heated to a really high temperature to sterilise it, to remove any bacteria, any microbes that might have been living in that agar jelly before we used it. We then split our plate, a bit like you can see on the picture there, and we had our normal hand as we'd come into the lab without any hand washing treatment, and we placed that onto the plate and labelled it up. Then we had the option of looking at using a hand sanitizer or hand washing, and we then placed our hand down again, and we can see the difference between that so-called dirty hand as we entered the room and the clean hand after we'd used our treatment. We're then able to compare our results to a friend who maybe tried the different hand cleaning method. So if you tried using soap and water and they tried a sanitizer, then you can compare results and see how effective those two methods were. This method can also be used, maybe you have some kind of infection in your body and they might take a sample from you, maybe a mucus sample, something like that, and then they can culture that to establish which antibiotics you should be given if it is a bacterial infection. Okay, so it can be used in lots of different ways, culturing that bacteria, identifying what it is, seeing which the most effective treatment might be. We used it in the hand washing method, but we can also use it to test the effectiveness of different cleaning products, maybe a more budget bleach compared to a more expensive bleach. So there's lots and lots of different ways that we can use this experiment to test different things. Hope you found that useful. Thank you.